Hello students, the topic of today's class is the electron transport chain, the third respiratory cycle in all living systems. We have already understood that all carbohydrates are converted to glucose and glucose is broken down to pyruvate in glycolysis. And pyruvate is converted to acetyl-CoA which goes through the citric acid cycle. The substrates or reducing equivalents like NADH2 and FADH2 that are formed during these two cycles enter the electron transport chain to yield ATP. We have also understood that lipids are broken down into acetyl-CoA which again go through the citric acid cycle, and the same pathway of the electron transport chain follows. So, today we will be studying the detailed reactions of the electron transport chain. The objectives of today's class is to understand the reactions of the electron transport chain, to understand the pathways that lead to the electron transport chain, and to understand oxidative phosphorylation leading to ATP formation. So, the contents are divided into 4 modules. The first module will deal with oxidative phosphorylation. We will understand what the term means. In the second module, electron transport that occurs through 4 complexes will be understood. In the third module, we will see how ATP is formed in the electron transport chain. In the fourth module, we will look at the different types of inhibitors and uncouplers of the electron transport chain that can cause this chain to be completely interrupted, leading to the death of the living organism. So, oxidative phosphorylation is basically the culmination of energy yielding metabolism in all aerobic organisms. All the oxidative steps in degradation of carbohydrates, fats and amino acids converge at this final stage of cellular respiration, in which the energy of oxidation drives the synthesis of ATP. So, the sites of phosphorylation is in eukaryotes, it occurs in mitochondria. And in oxidative phosphorylation, there is involvement of reduction of oxygen to water with electrons donated by substrates like NADH2 and FADH2. It occurs equally well in light or darkness. If we look at the proton transfer theory, our current understanding of ATP synthesis that occurs in mitochondria is based on the hypothesis that transmembrane differences in proton concentrations, that is across the membrane, inside the membrane as well as outside the membrane, there are differences in proton concentration, and these differences cause a transmembrane gradient, and this difference is the reservoir for the energy extracted from biological oxidation reactions. This theory was introduced by Peter Mitchell in 1961. This diagram shows you the electron transport chain across the mitochondrial cell membrane. The figure shown here depicts electron transport across the mitochondrial cell membrane. The cell membrane shown in white and yellow is a typical cell membrane which covers any cell or organelle. Here it is covering the mitochondrion, which has an inner membrane, an outer compartment and a inner mitochondrial matrix. The pink figures show the complexes or the proteins that transverse the cell membrane. They are called transmembrane proteins. Complexes 1 to 4 are the 4 complexes through which electrons are passed from NADH2 or FADH2. And once the electrons are passed, the protons are shunted out through each complex from the inner membrane to the outer compartment. And these protons travel to the last or the 5th complex, the ATP synthase, through which they flow back into the matrix and convert ADP to ATP. This in essence is about the electron transport chain. Let us look at each complex and the detailed manner in which the electrons are transported. This is a picture of the mitochondrion with the outer membrane which is freely permeable to small molecules and ions, and then you have an inner membrane which is impermeable to most small molecules and ions, including hydrogen, and which contains the respiratory electron carriers complexes 1 to 4, ADP ATP translocase, which is another protein which is attached to the final ATP synthase, F0 F1 ATP synthase as it is called, and other membrane transporters. Then the outer membrane is embedded with small ribosomes and porin channels to allow free movement of molecules. The matrix contains all the enzymes of the citric acid cycle, all the enzymes involved in amino acid oxidation, DNA and ribosomes, many other enzymes, ATP, ADP, phosphate, magnesium, calcium, potassium, many soluble metabolic intermediates, and the pyruvate dehydrogenase complex which converts pyruvate to acetyl CoA. So, this is the role of mitochondrion in metabolism because of the different compartments and the different complexes and enzymes that are located in different regions. Some important reactions catalyzed by NADP dehydrogenase or NAD dehydrogenase are indicated here. This is important because these reactions provide the substrates NADH2, NADPH2 and FADH2. In the citric acid cycle, the conversion of alpha ketoglutarate to succinyl CoA generates NADH2, malate to oxaloacetate which generates another NADH2, pyruvate to acetyl CoA in the pyruvate dehydrogenase complex, glyceraldehyde 3-phosphate to 1,3-biphosphoglycerate in glycolysis, lactate to pyruvate after glycolysis, then beta hydroxyacyl CoA to beta ketoacyl CoA in beta oxidation. All these reactions supply NADH2 as substrates to the electron transport chain. Then you have some NADP linked reactions where glucose 6-phosphate is converted to 6-phosphogluconate in the HMP shunt. Then you have L-glutamate which is an amino acid which is transaminated to alpha ketoglutarate which also generates an NADH2. You have isocitrate which is converted to alpha ketoglutarate in the citric acid cycle which also yields NADH2. So, this is where the NADH2s are generated in metabolic reactions. Now, we will talk about the different electron carriers like complexes 1 to 4 and their role in the electron transport chain. The mitochondrial respiratory chain, as it is called, consists of a series of sequentially acting carriers, most of which are integral proteins or integral membrane proteins with prosthetic groups, that is either metals or vitamins. In this case, the prosthetic groups are metal ions like iron and copper. These are capable of accepting or donating either 1 or 2 electrons. 3 types of electron transfers occur in oxidative phosphorylation. The first one being a direct transfer of electrons as in the reduction of ferric to ferrous ion, transfer of a hydrogen atom. As you all know, a hydrogen atom contains a proton with a positive charge and an electron with a negative charge. So, along with the transfer of each single electron of a hydrogen atom, there is also a transfer of a proton from the inner membrane space or the matrix to the outer membrane. And the third type of transfer is as a hydride ion which bears 2 electrons. So, the term reducing equivalent is used to designate a single electron equivalent transferred in an oxidative reaction or in an oxidation reduction reaction. Now, we will come to module 2 which describes the different complexes. The complexes are nothing but proteins and some of them are enzymes, some of them are small proteins which are not really enzymes. So, these complexes create a proton gradient across the inner mitochondrial membrane, meaning there is a lower content of protons on one side and a higher content of protons on the other side. This is called the proton gradient, and this is maintained by the action of the complexes of the electron transport chain, which consists of proteins associated with the inner mitochondrial membrane. The first one, or the complex 1, is nothing but an enzyme NADH dehydrogenase. As the name indicates, it is going to remove the hydrogens from NAD. Hence, it is called NADH dehydrogenase. In scientific terms, a reduced NADH is converted to an oxidized NAD by removal of hydrogens by the enzyme. The second complex, or complex 2, contains succinate coenzyme Q reductase or succinate dehydrogenase. It can be either of these two. Succinate dehydrogenase is an enzyme of the citric acid cycle. So, it can directly connect with the electron transport chain at that stage. The third, protein, it is not really a complex, it is part of complex 2. It is a protein which is part of complex 2. Understand here that a complex can contain an enzyme and a protein, or two enzymes or two proteins. Here complex 2 contains either of the two enzymes just mentioned along with coenzyme Q, also called ubiquinone. So, cytochrome BC1 complex is the complex 3, meaning it has cytochrome B and cytochrome C1. Then you have a small mobile protein called cytochrome C, which is associated with complex 3. And the fourth complex is the enzyme cytochrome oxidase, which is involved in the final transfer of electrons to oxygen to form water. Now, you see the location of the complexes. This is all complexes put together located on the inner mitochondrial membrane. The complexes shown in orange are complex 1, complex 3 and complex 4. Coenzyme Q and complex 2 are shown as two enzymes of complex 2 associated with the small protein coenzyme Q. Cytochrome C is shown as a small mobile protein associated with complex 3. And you can see the proton gradient with practically no protons in the mitochondrial matrix and a number of protons in the inner membrane space. This is called the proton gradient. So, in step 1, NADH, which has already been shown to be donated by a number of metabolic reactions of different pathways, binds to complex 1 and passes on 2 electrons to a flavin mononucleotide or FMN. This is a prosthetic group. As I told you, prosthetic groups can be metallic compounds or vitamins. Flavin mononucleotide is nothing but the B complex protein riboflavin. So, the FMN is reduced to FMNH2. So, whenever there is passage of electrons, you will see NADH2 has passed on its electrons and got converted back or oxidized to NAD. FMN has taken 2 hydrogens and got reduced to FMNH2. Then again ferric to ferrous. In that manner, the electrons are transferred and the protons which are part of the hydrogen ion will go from the inner membrane space, the mitochondrial matrix, to the inner membrane space. Electrons are then passed to the iron sulfur proteins FeS, which are present in complex 1. This is a non-heme ion. It is not associated with heme. It is an iron molecule or a ferric or a ferrous molecule independent of heme. The electron is accepted by ferric, which is reduced to ferrous. In step 2, 2 electrons from the reduced FeS proteins are then passed on to coenzyme Q or ubiquinone. When it accepts the 2 hydrogens, it gets converted to the reduced form. Coenzyme Q is thus reduced to COQH2, which is called ubiquinol. So, quinone has become quinol, while the iron sulfur proteins are oxidized back to ferric status. Now, what is the role of complex 2 in transfer of electrons? Complex 2 actually contains one enzyme, succinate dehydrogenase, which catalyzes the reduction of succinate to fumarate, is a reaction of the so called citric acid cycle. We have already seen the different reactions of the citric acid cycle, in which succinate is converted to fumarate with the help of succinate dehydrogenase. The same enzyme can be directly linked to the citric acid cycle through complex 2. In this case, NADH2 is disposed of. So, FAD oxidizes succinate to fumarate, FAD becoming reduced to FADH2 as it picks up 2 electrons and 2 protons. This FADH2 enters the complex 2. Succinate dehydrogenase is actually associated with complex 2 directly, and FADH is oxidized back to FAD by passing the electrons on to the iron sulfur proteins in complex 2. The electrons are then passed to ubiquinone or COQ, coenzyme Q, and are passed on to complex 3 through a number of iron sulfur proteins. Again in complex 3, they are passed on to cytochrome C, which is a small mobile protein. Complex 3 contains cytochrome B, cytochrome C1 and an iron sulfur protein. Like all FES proteins, cytochromes contain bound iron atoms, and this time the iron is a heme bound atom. The iron atoms alternate between +3 and +2, that is ferric and ferrous oxidation states, as they pass on the electrons. So, coenzyme QH2 passes on 2 electrons to cytochrome to cause the Fe3 plus to be reduced to Fe2 plus, ferric to ferrous, and the electrons are passed to the FES protein and then to cytochrome C1 ultimately. So, in step 4, cytochrome C is another small mobile protein, as I have already told, which accepts the electrons from complex 3, and ferric is reduced to ferrous again and shuttles them to the last electron transport protein chain, that is complex 4. In step 5, there is an involvement of complex 4, which contains both cytochrome A and cytochrome A3, both of which use iron and copper atoms to handle the electrons. 4 cytochrome C molecules pass on 4 electrons to complex 4, and these are eventually transferred with 4 hydrogens to oxygen to form 2 water molecules. So, if you look at the summary of the flow, there is an intermembrane space which is called the p side. In the first complex, NADH2, that is on the matrix side or the N side, is oxidized to NAD, and 4 protons are passed to the intermembrane space. Then in complex 3, another 4 protons are again transferred to the intermembrane space. In between, complex 2 converts succinate to fumarate and FADH2 to FAD. Coenzyme Q is also reduced to ubiquinol from ubiquinone, and ultimately in complex 4 you have 2 hydrogens passing from inside to outside. So, a total of 10 hydrogens are passed, and for 10 hydrogens you get 3 ATPs and 2 water molecules. So, in module 3 we will look at actual ATP formation. What has just been summarized is shown more in detail, and the plus charges in the intermembrane space indicate the highly polarized membrane because of the presence of protons. The proton gradient is having no protons or very few protons inside the matrix and number of protons outside the matrix. Now, these protons cannot remain outside, they have to flow back into the matrix to maintain an equilibrium. That flow is through the ultimate protein, the ATP synthase, the F0 F1 Atp synthase, which passes all the protons back through its channel into the matrix, and these protons induce the formation of ATP by combining with an inorganic phosphate molecule and forming ADP. So, the electronegative potential is being balanced by transferring the protons into the matrix and formation of ATP. This is the structure of the F0 F1 ATP synthase. It looks like a stalk with a tulip. The stalk is highly flexible and it is called the gamma subunit of the protein. You know, all enzymes are proteins which have different subunits, and then you have 3 alpha and 3 beta subunits which are like petals of a tulip. These are more rigid. Then you have the chamber in between shown in blue through which the protons pass. So, as the protons are passing through, there is a rotational change in different alpha and beta subunits which have sites for binding ADP and ATP. So, ADP binding sites are there in the beta subunits where ADP binds, and once the rotation occurs, this ADP accepts a phosphate, inorganic phosphate, and forms ATP. So, this is called proton pumping. Enough energy is released to pump one proton across the inner membrane as electrons pass from NADH through FMN to FES proteins of the complex 1. Electrons pass from cytochrome B to cytochrome C1 in complex 3. Electrons pass from cytochrome A to oxygen in complex 4. And finally, when FADH2 is oxidized by the electron transport chain, the first proton pump, complex 1, is bypassed since complex 2 has only enough reducing potential to pass electrons to coenzyme Q. So, this is how the proton pumping occurs. Ultimately, ATP generation occurs as indicated after the protons flow back into the matrix through the F0 F1 ATP synthase. Oxidation of NADH to NAD pumps 3 protons which charges the electrochemical gradient with enough potential to generate 3 ATPs. Oxidation of FADH2 to FAD pumps 2 protons which charges the electrochemical gradient with enough potential to generate 2 ATPs. So, although you will see these values quoted in many textbooks, recent information suggests that every NADH2 generates 2.5 ATP and every FADH2 generates 1.5 ATP. The reason for this is that not all of the energy stored in the proton gradient is used to generate ADP. Some of it is used to power the transport of ions in and out of the mitochondrion. Inhibitors tend to bind to components of the transport chain and block electron transfer. All components before the block will remain in the reduced state, and the components after the block will remain in the oxidized state. Clearly, no electron transfer is possible and proton pumping also stops. As a result, ATP synthesis also stops. The channel F0 of ATP synthase is completely blocked. Such inhibitors are clearly toxic as ATP is essential to all living processes in the form of energy. Some examples of inhibitors and what their action is are shown in this table. It shows that cyanide and carbon monoxide, which we all know are poisons, block complex 4 completely. Rotenone and amytal block complex 1, antimycin blocks complex 3. Oligomycin blocks proton channel F0 in ATP synthase. So, if you look at this figure, you will see that rotenone is blocking complex 1, NADH is not reduced. Antimycin is blocking at the site between cytochrome B and cytochrome C3 in complex 3. Cyanide and carbon monoxide are completely blocking complex 4 and water formation by transfer of electrons to oxygen. Now, there are also some uncoupling agents which will uncouple oxidation and phosphorylation. These make the inner membrane permeable to protons. So, there will not be any proton gradient which prevents ATP formation. The proton gradient leaks away, bypassing the ATP synthase, and electron transport has been uncoupled from ATP synthase. The energy released is wasted as heat instead of being stored as ATP. For example, the chemical 2,4-dinitrophenol, which is DNP, is lipid soluble, so it can diffuse across the inner membrane. DNP is protonated while it is in the inner mitochondrial space, since the pH is relatively low due to high proton concentration. As a result, it diffuses across the inner membrane into the matrix where proton is released, since the pH is higher. The proton has crossed the inner membrane and has bypassed the ATP synthase, and therefore, no energy or ATP is generated. Uncoupling agents also occur naturally. For example, in the newborn as well as in hibernating animals containing brown fat, the brown fat mitochondrion contain the protein thermogenin, which provides a channel through which the inner mitochondrial membrane is connected to the matrix. The heat energy released as protons rush down their concentration gradient through the channel, keeping the animal or the newborn warm. So, this is a natural process. To summarize, in the mitochondrion, you have a number of complexes through which electrons from reducing agents like NADH2 and FADH2 are transferred through 4 complexes to half oxygen to form water. In the process, protons are pumped from the matrix to the inner membrane space or intermembrane space between the inner and outer membrane. These protons flow back through the ATP synthase to convert ADP to ATP and form energy. And there are inhibitors which are present, which can inhibit this electron transport chain at various places from complex 1 to complex 3 to complex 4 to ATP synthase itself. So, these inhibitors are sometimes very deadly poisons like cyanide and carbon monoxide, which result in death of the animal or the human, because these inhibitors block electron transfer in intact mitochondria. Under these conditions, no ATP synthesis can occur and electron transfer to oxygen does not proceed. Coupling of oxidation and phosphorylation can also be demonstrated using chemicals or antibiotics like oligomycin and venturicidin that bind to ATP synthase in the mitochondria. These compounds are potent inhibitors of both ATP synthesis and electron transfer. When the flow of protons into the matrix through the proton channel of ATP synthase is blocked, no path exists for the return of protons to the matrix, and the continued extrusion of protons driven by the activity of the respiratory chain generates a huge proton gradient with no protons inside and a lot of protons outside. Certain conditions and reagents can uncouple oxidation from phosphorylation, and chemical uncouplers include 2,4-dinitrophenol and carbonyl cyanide p-trifluoromethoxyphenyl hydrazone called FCCP. After entering the matrix in the protonated form, they can release a proton, thus dissipating the proton gradient as heat. So, this is all about the electron transport chain and ATP formation. I am sure by now you understand the link between glucose oxidation, fatty acid oxidation, amino acid oxidation, tricarboxylic acid cycle, FADH2, NADH2 and the electron transport chain, ultimately releasing in synthesis of ATP or providing energy to the human system.